Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Amorphous Solid Dispersion, an ideal formulation approach to improve developability of poorly soluble molecules. My name is Sumaya and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending me a message using this chat panel. At this time, all participants are in listen-only mode. Please also note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Aragen, who developed the content for this presentation. Aragen Life Sciences is a leading R&D and manufacturing solutions provider for the life sciences industries worldwide. It offers end-to-end integrated or standalone solutions for small and large molecules. Established in 2001, the company operates through a network of sites located in India and the USA with a team of 3,800 plus scientists and 450 plus PhDs. Its expertise and experience have enabled over 400 customers to advance their research programs from discovery through commercialization. Aragen's innovative mindset, infrastructure, flexible business models have enabled them to serve large pharma, biotech, agrochemical, animal health, and performance chemical industries globally. Now, it's my pleasure to introduce our speaker for today's event, and that's Sreehari Babu. Sreehari Babu has over 25 years of experience in pharmaceutical development with 17 years in leadership roles covering early phase drug discovery support, product development in drug discovery R&D, generics formulation development, and clinical supplies management. Sreehari previously worked with Novartis Pharmaceuticals, Ranbaxy Research Laboratories, and Dr. Reddy's Laboratories. So now, without further ado, I'd like to pass control to our speaker. So Sreehari, whenever you're ready, you can go ahead and get started.
Good morning, good afternoon, good evening. Thanks for joining the webinar. I'm Sreehari. I will take you through my topic today. So the topic is amorphous solid dispersion, an ideal formulation approach to improve developability of poorly soluble NCE molecules, new chemical entities. So I put together the contents divided into three parts. I will briefly introduce you what is amorphous solid dispersion in this context. And I will also take you through the development process, how a typical solid dispersion is developed. And two case studies I will take you through in the end of the presentation. So the introduction. So just to give you the context of importance of amorphous solid dispersion. So in a typical NCE development phases, the main challenges or issues observed which causes the drugs not terminated or not developed further is mainly because of poor aqueous solubility, which results in a poor bioavailability and poor physical and chemical stability of the drug molecule itself, low permeability, first pass metabolism, and very short half-life, which limits its duration of action in the systemic circulation. So as you can see this from the literature, so drug discovery is a time-consuming and very expensive process. It starts with thousands of compounds being screened to hit the targets and eventually ends up into one FDA-approved drug over a period of 10 to 12 years time. So any improvement, even by small improvement of developability of even one more molecules may result into say 2 FDA approved drugs, which is tremendous improvement compared to success rate just by one more molecule. So this is that slide which shows the typical reasons for drug failures in the industry. So there are several reasons. Mainly the top two reasons are poor PK and ADME profile and lack of efficacy. Efficacy is more related to structure molecule itself. The second, the major reason of attrition is a poor PK admin profile, which also includes permeability issues. But the solubility issues can be addressed through right selection of right formulation approach. So this today's topic is mainly about how do we improve that, and discuss about the one of the major technique which is employed for more than 2/3 of the compounds in the industry. Use amorphous solid dispersion as a way to improve exposure, enable tox studies and clinical studies. So that these are the typical approaches, novel drug delivery approaches employed to address the solubility, bioavailability challenges. And the top choice typically is amorphous solid dispersion, which is mainly the converting the crystalline drug substance with very low solubility into an amorphous drug and embedded into a polymer matrix to stabilize it. And this is widely employed, and majority of compounds, and it works for any type of compounds without limitation. And the second approach is a lipid-based microemulsion formulation, suitable for highly lipophilic compounds, and also very highly organic content in terms of oils, surfactants, and solvents, which are not quite safe compared to the hydrophilic polymers we use in solid dispersion. And a nanosuspension, where the drug is still in a crystalline state with a particle size reduction to the nano size, increasing the surface area by several folds in a way to improve the dissolution. It doesn't improve the solubility as such, but it could be a good approach for certain kind of molecules. So today's topic mainly focused on amorphous solid dispersion, its key aspects of development and its challenges and its advantages and limitations, etc. So the overview of amorphous solid dispersion. So ASD is a dispersion of one or multiple drugs into an inert, hydrophilic, or water-soluble polymer at a molecular level in solid state. And the basic principle involves converting a crystalline drug, which is poorly soluble, into an amorphous form and stabilizing in a polymer at an appropriate drug load. And when this solid dispersion powder, or which is converted into a capsule or tablet, when this is exposed into an aqueous media in GI tract, the carrier dissolves first and the drug releases as a fine colloidal particles, still in amorphous state, forming a very high surface area, increasing the dissolution rate, upper end dissolution rate rapidly, and the polymer surrounded by polymer solution, which actually prevents the crystallization or precipitation of the drug, thereby improving the bioavailability for the duration of. So how the extent of supersaturation and inhibition of precipitation determines the how much improvement in the bioavailability can be achieved in the in vivo. And it's estimated that over the years that more than around 40 to 70% of NCEs are poorly water soluble. And within these, the majority of compounds are amenable to amorphous solid dispersion compared to other novel techniques. So this becomes a very critical technology which can help improve the developability of several molecules. So the advantages, coming to advantages of amorphous solid dispersion, is main advantage, it improves the drug solubility or dissolution compared to a crystalline drug, and thereby bioavailability and in vivo exposure. The vehicles used here are quite safe, the hydrophilic polymers, and a very small percentage of surfactants compared to the other liquid formulations. And improved wettability dissolution, and the drug is in amorphous state, stabilized in a polymer, and a very high degree of porosity of the powder. Limitations are, there are a few limitations, though it is a highly utilized technique to improve solubility. It does comes with some limitations. It's not suitable for compounds with a very high melting point if we go through high or very low solubility in organic solvents. So hot melt extrusion and spray drying are the very well-known technologies and commonly employed to commercialize and manufacture these solid dispersions. So if a compound has limitations in these two properties, this becomes very difficult to screen and develop a solid dispersion and also scale up and commercially manufacture. And the physical and chemical instability is always a risk in this type of formulation approach, as the drug is in amorphous state. And although we continue monitor carefully and control, protect from humidity, but this risk is always there for every solid dispersion, which requires extra monitoring, protection from moisture. And the complexity of manufacturing process, whether it is spray drying or hot melt extrusion, this limits the facilities available in each geography and then limitations. It's not as common as a regular tablet capsule manufacturing. That limits the options for companies to find a partner to manufacture GMP and supply both clinical and commercial facilities. So these are the next two slides are illustrates the typical techniques or processes used for manufacturing of amorphous solid dispersions, both at mini scale up and also at a commercial stage. The first process and most commonly used process and widely applicable to most compounds is the spray drying process, where the drug and polymer are dissolved in an organic solvent or a combination of organic solvents and sprayed at high temperatures using a spray drying machine. And this is a scalable manufacturing process. If the drug has appropriate solubility in a suitable and safer organic solvent at a reasonable solids content of 5 to 10%. It is a perfect process to manufacture for clinical and then commercial stage. And the second most widely employed process is melt extrusion, where the drug and polymer are melted at a very at elevated temperature, which is determined during the process development. Appropriate temperature where drug and polymer are miscible and also not degrading. And these at that particular temperature, the drug polymer mixture are extruded through a twin screw extruder and made into granules, which are further processed to make into capsules and tablets. This is also scalable manufacturing process and much more widely used in the commercial phase than the clinical phase due to material requirement at the clinical stage. And it's not suitable for temperature sensitive compounds. So it's more limited compared to the solvent evaporation process in terms of applicability to the type of molecules. So the other three techniques, which are typically employed in a research phase and the screening phase, but not so much employed in the GMP manufacturing and commercial phase, are the lyophilization, where it's all solvent evaporation processes. Lyophilization involves the same as a spray dried solution prepared in a freezing low freezing point solvents and freeze dried using a lyophilizer to remove the solvent completely. And this is suitable for screening and a small scale up size, but not really amenable for very large scale up processes. And co-precipitation, where drug and polymer are dissolved in solvent and precipitated with the help of an anti-solvent, very similar to, and then solvent is evaporated later. So this is also good for screening and the research work and not much utilized in the industry for clinical and commercial scale. And the last one is supercritical fluid technology, where drug and polymer are dissolved in a solvent and and carbon dioxide is used as an anti-solvent to cause rapid precipitation. So these are three other technologies which can be utilized for screening and quick development, because it's not feasible to do spray drying in the screening due to the compound limitations. So these are ideal for early stage proof of concept development work. So that's the introduction of ASD and how it plays an important role, and the background of the technology, the pros and cons of the ASD. So next few slides, I will take you through the development process, how it is, how ASD is developed typically in the industry. So the key considerations in the process of screening, scale-up, process development are starts with the polymer selection. And in the second screening, we select a combination of polymer and surfactants, and we consider and evaluate drug polymer miscibility of the selected polymers, and we need to optimize the drug loading, usually between 10 to 40%. 40% tend to be upper limit where we can achieve stable solid dispersion. In vitro kinetic solubility or dissolution in a bioavailable media, and in vivo evaluation in rodents and non-rodents PK studies to confirm the improvement in the bioavailability. And once the formulation is confirmed, selected, composition is selected, confirmed in the in vivo studies, then manufacturing process is developed, either spray drying or hot melt extrusion, and which is transferred to a GMP and then supplied to clinical studies. That's the process. It requires a lot of both solid state knowledge and also formulation knowledge to be able to understand how the drug and polymer coexist, and then able to predict the long-term stability, physical stability of the mixture selected composition. And this slide shows the typical polymers and surfactants employed. Though there are other polymers, but these polymers and the surfactants are typically used in the industry. They have been used in several safety studies and clinical studies. So these are the first go-to choice. And the process involves screening these polymers at a certain drug load to identify the best polymer, 2 or 3 polymers, which have improved. The first consideration is polymer will convert into amorphous form. And the second consideration is, does the solubility improve over the drug substance or drug itself? And then these are the typical surfactants utilized in very small quantities, 2 to 5% levels, compared to which are at a safe level. So the first step in the entire development process is the screening at a very small scale, in a milligram scale, both polymers and surfactants. And the main criteria selecting the best combination is drug is in amorphous state, and kinetic solubility in SCF and phosphate is improved over the crystalline free drug by as high as we could achieve. So that's the criteria to select the best polymer and surfactants, and then combination, and they're further screened in multiple levels to optimize the drug load before we go into the scale up stage, where the scale up, few formulations are selected for the scale up, optimized this is not yet process developed, it's only a scale up at a very small scale. And these formulations are thoroughly characterized in terms of solid state, solubility, chemical and physical stability, and also PK studies in different species to confirm and select the best one formulation, which would be used for process development, manufacturing process development. So this process would take typically 4 to 6 weeks, and then more than 100 combinations in terms of polymers, surfactant, drug loading, etc. in a variables are screened to lead up to the one best formulation, which can be developed further into process and then manufactured under GMP. So the screening involves freeze drying and scale up or the manufacturing process typically involves spray drying or hot melt extrusion. The characterization of coming to characterization, only the final scale up, selected 2 or 3 formulations are thoroughly characterized to understand the best formulation to go to PK studies. So typical characterization includes the powder X-ray, modulated DSC, TGA to understand any solvent presence, and hygroscopicity by DVS, water content, particle shape and size by SEM and PLM. And drug and polymer interactions are studied through FTIR or Raman to understand if there is any interaction or their physical coexistence in the matrix. And the residual solvents estimation by gas chromatography, and physical and chemical stability at long-term and accelerated conditions with the protection of humidity and without protection. And the last important piece of the data is in vitro kinetic solubility and dissolution in biorelevant media. So once all these are confirmed, the ideal solid dispersion must be stable, improved solubility over the neat drug, and has acceptable TG value, typically above 60 degrees, and the drug should be in amorphous state. Those are the criteria based on which formulations are selected for the PK studies. So the last piece of the development is process development. Usually spray drying is the go-to process because of minimal compound requirement and ease of process. And in some cases where the solvent solubility is so low for the compound, one may choose a hot melt extrusion as a choice, but which requires typically at a clinical phase a lot of material to have optimize the temperature, develop the process. So this is the R&D scale mission. Sorry, this is a typo here. This is 1 kg per hour drying. And this is the first process development where we can do at 100 gram to 1 kg level process optimization, which is typically used for tox studies, supplies, and develop R&D scale process to convert into a tablet or capsule. And then eventually the process is transferred to a GMP spray dryer where the batch is manufactured under GMP and then converted into tablets and capsules to supply to clinical studies. So that's the development introduction and development. The last phase of the this presentation is I'll take you through a few case studies. So that one first case study involves the development of amorphous solid dispersion for a neutral molecule. They're all NCE molecules, whatever I'm discussing here, and with the objective to improve oral bioavailability. The compound is crystalline in nature with a melting point of 203 degrees centigrade, and the solubility in FASIF is around 2 microgram per ml, and oral bioavailability in rodents and dogs are less than 10%. This is the objective and also the challenge. Mainly to challenge is to improve the FASIF solubility and thereby improving the oral bioavailability to several folds, so that the molecule can be taken to tox and clinical studies. So our standard approach, we have standardized the protocol and then processes, though the final composition customized to each molecule. The initial screening process are quite standardized. So we develop by screening with polymers and surfactants to identify a suitable composition with improved in vitro kinetic solubility, acceptable stability profile, and develop the manufacturing process for scale up studies. So a stable solid dispersion was successfully developed at 20% drug load, and the formulation showed good chemical and physical stability over 1 month at accelerated condition, and the in vitro solubility showed over 50-fold improvement, which doesn't exactly lead to that many fold in the in vivo, but usually the solubility shows much higher improvement than the in vivo. And the in vivo exposure in the dog PK increased over 5 folds compared to the crystalline micronized suspension. So these are the molecule's typical properties, solubility property. As you can see, the solubility is almost minimal, 2 to 5 microgram per ml across the pH range. It's more like a neutral molecule. So the challenge is mainly in FASIF, how to improve the solubility to a desired level. So this is an example of, I didn't list all the development activities we conducted, but this is an example of listing the typical polymers we utilize and also the solubility, how we in vitro kinetic solubility, how do we conduct by HPLC methods. So these are typical polymers, and we convert them to, we screen them through freeze drying process and tested them for XRD to confirm they're all amorphous. In all the cases, drug is in amorphous state. However, drug is in amorphous state, but the solubility, as you can see, is quite different between polymer. So the amorphous drug nature alone won't be adequate to improve the solubility to the desired level. So hence the polymer plays a key role, and sometimes surfactant as well. So from this list, as you can see, that Soluplus and copovidone showed the best improvement in the solubility compared to most other polymers, mainly considering the solubility in FASIF, with Soluplus being the best and closely followed by copovidone. So we pick these two polymers and combine with them with increase the drug load. As we increase the drug load, usually the solubility gets compromised. And then that's when we include a surfactant to hold the supersaturation further and make several combinations until we are able to stabilize the solubility levels at a higher drug load. So this is an example of final selected formulation in vitro kinetic solubility. So solubility is typically targeted at 2 milligram per ml. And as you can see, micronized suspension almost has no solubility. As we see in equilibrium solubility, it is correlating with that. And the selected amorphous solid dispersion is improved the solubility in SGF and then maintained the supersaturation post that in FASIF. It's a two-step dissolution model in the same conditions. We just add the FASIF to the SGF and observe whether drug is still in solution state or precipitating. And this formulation is typically subjected to rodents and non-rodents PK studies. And this is the dog PK data illustrated here over a 24-hour period. And so we see over 5 to 6 folds of bioavailability or oral exposure improvement compared to the micronized suspension, which is typically adequate. As we increase the dose, it may go up to, dose proportional increase could be observed up to a certain dose, and which usually enables the molecule to complete the tox studies at a desired exposure levels and also enable the smooth clinical studies with guaranteed exposure at the clinical doses. So the second case study is very similar to the first one, but a different type of molecule. It's a weakly basic molecule with pH-dependent solubility, though the solubility is quite good in low pH at acidic pH due to it's not a biorelevant media, not absorption site for many compounds, still shows almost very low bioavailability, just like a neutral compound where there's no solubility across the pH range. So this compound is also crystalline in nature with a melting point of 255 degrees and solubility in FASIF of about 5 microgram per ml with oral bioavailability less than 5%. The main issue with these kind of compounds they dissolve in low pH and then crash out, precipitate out rapidly at higher pH after pH 4 and above. So that causes very little absorption. And then especially in preclinical species where the pH are typically higher, in those cases, these drugs won't even dissolve in the first phase, and that affects the overall viability of this compound. We conduct the same screening, but it ends up into a different, based on the molecule, different polymers are selected and surfactants and the drug load. So it's at this stage, we can see that we typically optimize all the formulations at 20% drug load at the tox phase, and then we, as the clinical doses comes down, we optimize this further to improve the drug load to up to 40% at the later phase. So this, in the next slide, the solubility, you can see that at pH 1.2, 2, and 3, there is a decent solubility, which is good enough. But however, at higher pH, including in bioavailable media at high pH, the solubility is almost negligible, which is mainly the precipitation or supersaturation issue with this compound. The solid dispersion main objective is to prevent the precipitation and maintain the supersaturation at higher pH for these type of molecules. We conducted the polymer screening for this molecule, and as you can see that it's only only one polymer works best. The second best was a new polymer called Amorphous. It worked for some time, but we typically, as a first choice, we go with the well-known safe polymers with the known tox data. And in this case, there's only one polymer which improved solubility to more than 100 folds in FASIF. And we further optimized it, increased the drug load, and optimized with the combination of surfactants, and conducted the same solubility studies. So here you can see that micronized suspension also dissolves itself in low pH due to its inherent solubility of the crystalline drug. But when we change the pH, it rapidly precipitates, simulating the GI tract, the event, how it happens when the dosing happens. And the amorphous solid dispersion with the help of polymer mainly prevents the supersaturation or precipitation. It's not only the solubility improvement, but maintaining the supersaturation so that drug is in amorphous state and soluble and absorbing within this window. And we conducted dog PK studies for these molecules, as well as you can see that micronized suspension, mainly because of precipitation effect, has very low bioavailability, and solid dispersion showed very high Cmax, and then followed by sustained, because of polymer, the exposure is sustained over a long period to almost 3 days. Which actually improves the AUC and exposure even more because of sustaining effect of the polymers. So this is the overall summary of what we discussed so far. So amorphous solid dispersion involves strong experience and skills in terms of screening, selecting a polymer, understanding the drug polymer interactions, miscibility, the TG values, which are important for future. Otherwise, low TG compounds will create a lot of issues in terms of sticking, spray drying, or turning into gel form when it is compressed, etc. So selecting a right combination of formulation which has optimal solid state properties and also kinetic solubility is critical. So solubility is not the only criteria while we select a right composition. And in the second phase, the expertise involves is the process development, selecting appropriate process for scale up, and followed by the typically we employ dry granulation for amorphous solid dispersions, followed by compression or capsule filling, depending on the dose of the molecule for clinic. and film coating if it is a tablet. So basically, we convert the crystalline drug substance into a amorphous solid dispersion tablet, and manufacturing from and screening to clinical supplies, and then commercial beyond. So very robust technique, though very complex process in terms of regular tablets and capsules. But with the right expertise and experience, this also can be simplified and developed in the same timelines as the conventional tablets or capsules. So yeah, so this is my last slide. So please feel free to contact us if you need any help or guidance on resolving solubility of any molecules. So thanks for attending the webinar and listening to me.
Well, thank you very much for that insightful presentation. So audience members, that does bring us to a Q&A portion of today's webinar. So if you have questions for Sreehari, keep sending them in. I've already received some questions, so let's go ahead and start off with those. So Sreehari, the first question here is, what is the typical timelines of ASD development?
Typical screening takes around 4 to 6 weeks, followed by process development for another 2 to 4 weeks. So it's typically takes about 8 weeks to complete screening to process development. And then there will be additional 6 to 8 months to develop and convert into a clinical formulation like a tablet dosage form.
Thank you for that answer. And another interesting question from an audience member. That question is, what are the alternative techniques for molecules not amenable for ASD?
So as I listed in this slide, the typical techniques used for solubility improvement, which are scalable to commercial scale, are microemulsion and nanosuspension, which are suitable for molecules. For example, microemulsion is suitable for a molecule which has high lipophilicity, log P, and nanosuspension is suitable for molecules which can maintain their crystalline state under milling, in intense milling activity. So they're not as amenable as solid dispersion for most molecules, but they are an alternative if in any case solid dispersion doesn't work for a given molecule.
Great, thank you once again. Our next question here from an audience member is, does Aragen offer complete ASD solutions from screening to GMP manufacturing?
Yes, so I covered that in the previous slide. So yeah, we do offer as early as drug discovery phase, where we get molecules from discovery chemistry of very small quantities of molecules between 2 to 5 grams. We can do a screening at a very miniaturized scale through solvent evaporation process, and take that approach screening outcome into all the way up to GMP manufacturing at our facility in Hyderabad.
Thank you. And our next question is, what is approximately the upper limit for MP for ASD?
Sorry, approximate NP means drug load.
I can ask the attendee to explain this further if you like, or and we can go to the next question.
Yeah, I just, can you repeat that question? Maybe I would try to understand.
Sure. So it is, what is approximately the upper limit for MP for ASD? And I also see the attendee has put in maximum percentage.
Yeah, drug loading percentage. Yeah. So typically it varies between compound to compound. Maximum typically is 40%. Ideal is 25 to 30%, where we have a balance of stability versus solubility improvement. Beyond 40% compounds which can improve solubility beyond 40% usually don't require such complex formulation approaches, because there is not enough polymer to stabilize the drug in amorphous state beyond 40%.
Great, thank you for that answer. Another great question coming in from an audience member. That question is, does surface area or molecular weight of polymer has any effect on solubility enhancement at higher drug load?
Yeah, we didn't see surface area as playing a role, but we do see molecular weight does have a role sometimes on maintaining supersaturation for a longer period. Mainly because the polymer, when it dissolves in the GI fluids, it maintains a higher viscosity than the other polymers, which in turn prevents the drug precipitating out. So that's the main cause of it. So the molecular weight does play a role, but may not be the particle size of the polymer, because when we dissolve and spray dry, it becomes into a different particle size completely from the input polymers.
Thank you again. Another great question from an audience member. The question is, in amorphous solid dispersion, how stable are the polymers and surfactants? Is the combination of polymers and surfactants create any instability of the drug because it involves the heating?
Yeah, so for the drug which is stable at a certain temperature, suppose for spray drying, we do it typically at 60 to 100 degrees temperature, inlet temperature. So the drug must be stable at that temperature for short periods of duration, and it should be stable in solvent. That is to handle the processability point of view. And then the drug also should be compatible with the selected polymers. In a rare cases, there is a suppose acidic drug and we select a basic polymer, which may have some interaction, chemical interaction, which can probably degrade the drug substance. So in some cases, but most polymers are neutral, they don't cause any chemical stability issues, but there are exceptions we observed. So the stability study or the compatibility study would clear those polymers out. So if we have a excellent solubility with that polymer, we still may not be able to select due to stability concern. And surfactants actually improve certain polymers which are not very highly soluble, like enteric polymers, etc. Surfactants can actually help them to be wettable and push to them to dissolve faster compared to a simple polymers like povidone or something which are hydrophilic, they dissolve easily. So that's why we use very small percentage of surfactant to fast track the dissolution rate of certain type of polymers. They can cause physical instability, making the amorphous drug molecule particles more mobile and then try to crystallize. But it's a right combination of polymer and surfactant percentage, we can stabilize that system.
Great. Well, thank you very much for all of those answers. We've reached the end of our Q&A session. So attendees, if you do have more questions, you can keep sending them in using the contact information on your screen. So that's info@aragen.com. So go ahead and please jot this down. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Additionally, I've just sent a link in the chat box to view the recording of this webinar. So that should be uploaded in the next 1 to 3 business days. And if there's any colleagues or anybody who might be interested in the webinar, please go ahead and share that as well. Now, please join us in giving a big thank you to our speaker today. That's Sreehari Babu for that fantastic presentation. We hope you found this webinar informative. Have a great day, everyone.
Thank you very much. Bye.